Hi, my name is Bhumika, and the lesson for today is going to be beta oxidation of fatty acids. Okay, I'm actually going to make the structure of fatty acids in a different presentation altogether. So this is only going to be the cycle of the fatty acids, which is beta oxidation. Overview: in the 1904, Franz Newt proposed that fatty acids are degraded by the sequential removal of 2 carbon units from the carboxyl ends of the molecule. The degradation, it takes place by oxidation at beta carbon. Most important. David Green, Severo Oka, and Fyodor Leinen, they made important contributions to the understanding of the series of the reactions, which is called the beta oxidation pathway. Now, I have drawn all the cycle because I think it's more easier to understand it this way in more of a diagrammatic position. Okay, for the first step, we've got activation. Now, a simple structure of fatty acid, I have given it here. It is a long chain of hydrocarbons with an R group, which is any number of carbon, alright, and a carboxyl group onto the other terminal. Now, this, by utilization of 1 ATP, forms acyl adenylate. And this acyl adenylate, in presence of an enzyme called the acyl-CoA synthetase, forms acyl coenzyme A. Now, this acyl coenzyme A, it undergoes all the further processes of beta oxidation. Now, this whole process, it takes place outside the mitochondrial membrane. So we've got a protein known as carnitine, which attaches itself to this acyl CoA, and it forms acyl carnitine. Now, this acyl carnitine, it shoots through the inner mitochondrial membrane, as I've drawn this here, and by addition of a CoA-SH bond, it separates into an acyl CoA and carnitine. This carnitine then again goes outside the inner mitochondrial membrane, and what happens? It reutilizes, and it combines with more acyl CoA and forms acyl carnitine, and then brings them back inside, and as such this whole cycle it goes on. Alright, acyl CoA, as I told you, it undergoes all the further steps. Now, a simple structure is drawn here. The first step is oxidation. Now, this oxidation requires an enzyme known as acyl CoA dehydrogenase. What happens is, here at the beta carbon, hydrogen is extracted, and as such a double bond is produced, as you can see. So now here 1 FAD plus, it forms or converts into FADH2, which is a very high energy carrying molecule. For the next step, the enoyl coenzyme A, so formed, undergoes hydration. That means simply addition of water, H2O. Now, this takes place through the process, through the enzyme, which is known as the enoyl-CoA hydratase. And as such, the double bond, it breaks up, and OH and H is attached. So we can see the formation of a new hydroxyl group. So this is the second step, which is the hydration. For the next step, again oxidation takes place, and when this step takes place, 1 NAD plus forms NADH, which is again high energy carrying molecule, and the enzyme which is responsible for this change is the hydroxyacyl CoA dehydrogenase. Alright, now you can see that this OH group converts into a double bond O. So we've got a new carbonyl group at this position. Okay, now if you notice, this side, it basically represents the acetyl part, whereas this side we've got an acyl part. So the next step is called thiolysis. In thiolysis, breakdown takes place right at this position that I've drawn. Okay, after this, in presence of the enzyme thiolase and addition of CoA-SH, it forms acetyl coenzyme A, which enters the TCA cycle or the Krebs cycle, and it also forms an acyl coenzyme A molecule, which is 2 carbon less than the initial compound. So as such, it goes the cycle again, and we already have got our product acetyl coenzyme A, which is the substrate for the TCA cycle. To sum it up, we've got the first step, which is activation. Then we've got the second step right here, which is oxidation number 1. The third step is, of course, hydration. The fourth step is oxidation number 2, and finally the fifth step, pyrolysis. So over and out, it produces acetyl coenzyme A as its final product, which acts as a substrate for the TCA cycle. Thank you for watching this video. That's it for today's lesson.